So in the last video, we were talking about the structure of DNA, how we get DNA to fit into cells, how the two strands of DNA hold together, the overall structure of DNA, right? The physical characteristics of DNA. Now here, we're going to start talking about how DNA functions in cells. This video will focus on transcription, the process of transcription. But really, this video and the next video, translation, we're going to talk about how the information in DNA is used to make all of the stuff that is you, that is me, that is every living organism. How is DNA used as an instruction manual to build all of those molecules? Okay, so again, DNA is a hereditary information, but really what DNA is doing, it's containing the recipes, the information for how to make proteins and other molecules. Right? How to make proteins, how to make fats, how to make phospholipids, all of the molecules that are important for a cell to function. DNA has the information for how to build those. And DNA has many genes. Our genome has many hundreds of thousands of genes in it. Remember, a gene is just a segment of DNA, just a length of DNA that contains the instructions for how to build a specific protein or a specific molecule, right? So we're gonna watch the transcription and translation of a gene here, okay? So because DNA contains the information for how to build all of the molecules necessary for life, all of the proteins, all of the phospholipids, right? If we change that recipe, if we change that DNA code, the sequence of letters, the A, T, C's, and G's in DNA, that could result in a change to the overall molecule that results from it. A change to the structure of the protein, which, as we know, if you remember nothing else from this class, protein structure determines its function, right? So we change the DNA, that could result in the structure of the protein changing, which would mean that protein may not function properly, right? And so we call changes to the DNA sequences mutations. Okay, so mutations can be harmful to an organism. So what we're doing here is, yes, we need to use that recipe that's in DNA, that information that's in DNA, but instead of using it directly, instead of reading it directly, we're going to just copy off the pieces from that recipe book that we need, and then take those pieces out, those copies out, in order to use those as instructions. I like think of DNA, so like I have some old handwritten recipes from like my great-great-grandmother. They're on this like really yellow paper, it's like cardstock, it's all written in her really old-timey cursive, right? But they're really special to me because I actually knew my great-great-grandmother. It's a whole other story, but they're really special to me. They're like family recipes, family heirlooms kind of, right? So I'm not going to take out that recipe card, that original recipe card, take it into the kitchen with me and use that recipe card as it is. What I do, I take a picture of that recipe card and then I cook from the picture, right? Why don't I use the original recipe card? I don't want to damage it. I'm a messy cook. Stuff goes everywhere, right? I don't want to accidentally set it on fire, which I just set something on fire the other day on accident, right? So I don't want to damage that original copy. So instead I make a copy of the original and I use that copy of the original to cook, right? And that's kind of what we're doing here with DNA. Where DNA is the original copy, we're going to make a copy of the original and use that as our instruction manual to build these proteins. Here, the copy of the original is going to be a molecule called messenger RNA, mRNA. We've talked about this briefly before, but messenger RNA is just an RNA copy of a segment of DNA, of a gene. Okay, so messenger RNA is going to contain the information necessary to build that protein. Okay, but it's just that one specific segment of DNA that we're going to make a copy of in RNA form. And that RNA can leave the nucleus. Remember, DNA is hanging out in the nucleus. It's safe in the nucleus. That RNA can leave the nucleus and hit up a ribosome, say, hey, I have a message for you. Let's build a protein. And that's basically how it works. So we call this kind of process of DNA original instructions. We get a copy of DNA in the form of RNA, and then that RNA is what's used to build a protein. We call this kind of like biology's central dogma. It took us a while to figure this out. We didn't really know exactly how this functioned, but once we did, we're like, whoa, that's how it works. Okay. DNA is the original, RNA is the copy, protein is the final product, okay? So this first step, making a copy of DNA, copying the DNA and transforming it basically into an RNA copy, we call that transcription. So like right now, you're probably taking notes. Your notes are very similar to what I'm saying, but you're not writing down every single word, right? So you're transcribing the important information. That's basically what's happening here, from DNA to RNA. We're taking just that gene segment of DNA and making an RNA copy of it. Okay, so that's transcription. The next step is turning this RNA copy into a protein, into a physical protein that can do stuff, like break down lactose, or maybe it's a protein that's on the surface of the cell that's used for signaling, right? So we take this information that's an RNA, that's information that is the sequence of bases that are in this RNA, and we turn it into a protein product, amino acids, right? That process is called translation. So DNA to RNA, transcription. RNA to protein is translation. Those are the names of those processes. Okay, transcription. So transcription, we find a piece of DNA, we're going to transcribe a gene into mRNA, messenger RNA. That's our big picture here. That's what's happening. We're going to transcribe a piece of DNA called a gene. So a gene is a sequence of DNA that contains the information for how to build a protein. We're going to transcribe that gene into an RNA copy of it. So we're in the nucleus, because that's where DNA is at, right? And we're going to talk about the major steps that's happening here in transcription and the enzymes responsible for what's going on in transcription. So these will be the same steps: initiation, elongation, and termination. We'll see these same steps again when we talk about translation. Okay, so that might be a little confusing. Initiation, elongation, termination. We have those three basic same steps in both transcription and translation. But what I want, by the end of this, you should be able to describe this process, explain to me which enzymes are being used, and you yourself, if I give you a piece of DNA, a sequence of DNA, you should be able to transcribe it. Okay, so what does this look like? So our main enzyme player here, you should know this, our main enzyme player in transcription is a molecule that we call RNA polymerase. So polymerase is just a class of enzymes, and really they're just builders. They are the ones that are constructing longer molecules. They're the ones constructing polymers. This one, based on its name, RNA polymerase, is building RNA. Okay, so polymer, remember, long strand of monomers. ACE, enzyme, and RNA tells us what type of molecule is it building, what type of polymer is it building. So RNA polymerase is the enzyme that will read the DNA, it will read the original gene on the DNA, and make a copy of that gene in the form of RNA. Okay, so it's going to read the original DNA and find base pairs that match up with that DNA to make an RNA copy of that DNA. This usually all happens at a region called the promoter area. It's a region just upstream of the actual information of that gene. So it's a region that says, okay, here's the doorway, here's where the gene starts. So RNA polymerase, you need to bind here on this DNA molecule. And it's a repeating sequence. This promoter region is usually a repeating sequence of TA, so we sometimes call it the top box. But really it's just a signal for where does RNA polymerase need to land? Where does it need to start? Because RNA polymerase doesn't know where to go. It has to have a substrate to bind to, right? So what's going to happen is RNA polymerase is going to bind to that DNA strand. It is then going to separate those two DNA strands. It's going to break the hydrogen bonds between the two DNA strands, create an RNA molecule that is complementary to the DNA. So, right, the two strands of DNA are complementary. They're not identical, because if one strand we have an A, on the other strand we have a T. So they're complementary, but not identical. Okay, this is just a diagram of what's happening. So here's our DNA strand. Okay, this is our sense strand. This is the one that is being transcribed. So we call this strand the sense strand. And the other strand, the non-template strand, we call this the nonsense strand, basically. So RNA polymerase, here's our promoter. This is our top box. Remember, this is just a region of DNA that says, hey, RNA polymerase, this gene starts here. It's basically the start location. RNA polymerase is represented by this kind of beige enzyme right here, right? This blob, but it's an enzyme, and basically what's happening is it is bonded to the DNA, and it's reading the sequences of bases here on the DNA and matching them up with a complementary RNA base, right? So let's say we find on the DNA we have a T here. On the RNA, the complementary base would be an A. We have a G on the RNA. If we have a G on the DNA, the complementary base on the RNA would be a C. Okay, so it's just using this as a template for how to build this RNA molecule. So it's going to bind to the promoter, and then it's just going to kind of zoom along until it reaches a stop signal. So we have a start signal at the promoter, we have a stop signal later on downstream. But all of this stuff between the promoter and between the stop signal, between the promoter and the stop signal, this is all DNA that contains information for how to build a protein. All right, so here we're looking at it a little bit more in detail. Okay, so here's our DNA template strand. This is the one that contains the information for how to build a specific protein. Here's the complementary strand of DNA in blue here. Here's our RNA polymerase, right? So RNA polymerase, our promoter would have been back here. RNA polymerase moves along DNA in the 3' to 5' direction. So RNA polymerase can only move in one direction. So it starts at the 3' end and moves towards the 5' end of DNA. Like, holy crap, I forgot what that means. What does 3' and 5' mean? Hang on a sec, I got you. So 3' and 5', remember this is what it means. In our sugar, there are 5 carbons. We number those carbons moving clockwise from the oxygen. So 1, 2, 3, 4, 5' is this one right here. 1, 2, 3' is this one right here. So it just gives a directionality to the molecule, right? So remember, if we look at DNA, like this one we looked at before, this is our 3' side, this is our 5' end. But because they're running upside down, they're running anti-parallel. If on one strand of DNA we have the 5' facing up, the other one's going to be upside down. It's going to have the 3' facing up, right? So our 1, 2, 3' carbon right here, 4, 5'. So this end is our 5'. So it's just a directionality to that DNA molecule based on the orientation of the nucleotide. So RNA polymerase can only move from the 3' end towards the 5' end because of the shape of these molecules. It cannot recognize them moving the other way. It can only move 3' to 5'. But what that means is that because it's anti-parallel, that means that RNA polymerase will move along the template DNA from 3 to 5, but the RNA it constructs is 5' to 3', right? Because it has to be complementary and they run anti-parallel. So it reads DNA 3' to 5'. The RNA it builds is 5' to 3'. Okay, because they have to be opposite basically. Okay, so look here. All right, so here we have an A in the DNA. The complementary RNA nucleotide is a U. Remember, because there's no T in RNA. Here's a T in the DNA. The complementary RNA is an A. G, C, C, G, right? So build a complementary RNA copy of that DNA sequence. So it's going to continue moving along, opening up the DNA, breaking these hydrogen bonds between the two strands of DNA, continues moving along, breaking those hydrogen bonds until it hits a termination sequence. So just like we had a promoter sequence, right here that told RNA where to start, we're going to have a termination sequence at the other end of the gene. So RNA polymerase is going to continue to elongate and continue to build this RNA copy until it hits a region that we call the termination sequence, which is really just a region kind of similar to Tata box. It basically just tells the RNA molecule, once you hit this sequence of bases, that is the end of the gene. Okay, so it's basically just telling RNA polymerase, that enzyme, that, all right, this gene is done, you don't need to keep transcribing. Okay, that's what happens when it hits the termination sequence. So it's just a sequence of DNA that says, RNA polymerase, you're done. This gene is finished. So now we have, at the end of transcription, we have an RNA copy of this DNA sequence in here, which is pretty cool. It basically made a photocopy of just that one sequence of DNA that we needed. We don't need all of the genes in DNA all the time, right? We might only need this specific gene for a little bit of time. So we can just make a copy of the ones that we need in the form of RNA. RNA is single-stranded, it's cheap, it's easy to build, so we're going to make a copy of RNA. So now we have a single-stranded copy of a gene in the form of RNA, in the form of an RNA transcript. Okay, now I'm going to switch over to my tablet and we're going to practice transcribing a gene. Sound good? Let's practice this process of transcription. We're going to practice this process of transcription. So remember, transcription, we're moving DNA, making a copy of DNA in the form of RNA. Okay, so I'll do DNA here in purple. So let's say we have a sequence of DNA. Okay, so here's our DNA. Here is our 3' end. Here is our 5' end. Remember that 3' and 5', that is just the orientation of that molecule, of our nucleotide, right? So on the other strand of DNA, we're going to use this one as our template strand. This one here is going to be our template strand, right? The other strand of our DNA would be running, though, 5', 3'. Okay? And the reason why that's important is because enzymes can only read or interact with DNA or RNA in specific directions, right? Because the active site of that enzyme is specifically shaped. It has a very unique shape and chemical environment. So if it reads 3 to 5, it can't read 5 to 3. It can't do that because it's a totally different molecule, basically looking the other way. It's a totally different orientation of all those atoms in the molecule. Okay, so again, here's our DNA in purple. I'm going to do RNA now in red. Okay, so if you haven't already, write down the sequence of bases and then pause and try and transcribe this gene first on your own. Okay, so first of all, let's figure out which direction. So if we have a 3' over here, the RNA we're building, the 5' would be here, and the 3' would be here. Again, because they are running anti-parallel. DNA and RNA, whenever they make two strands like this, they are running anti-parallel to each other. Okay, so meaning one is running 3' to 5', the other is running 5' to 3'. Okay, they're running basically upside down from each other. So now we just got to figure out what are the complementary bases of RNA that would match up with this DNA molecule. So usually in DNA, we have A and T bond together, C and G bond together. What's different about RNA? In RNA, remember, there is no T. There is no T. Instead, we replace that T with a U. So A and U paired together and C and G paired together in RNA. Okay, there's a T, it's a DNA. There's a U, it's an RNA molecule. Okay, so A in DNA would pair with T, but we're in RNA, so it's going to pair with U. T pairs with A. C and G. There we go. Cool. So now we've just transcribed our first strand of RNA. So we just transcribed a piece of DNA into RNA. Let's go back and see what happens to that RNA after transcription is done. After we've made that RNA copy of DNA, let's see what's happening to that molecule now. We're going to process that RNA transcript before it will be used to actually build a protein. Okay, so we're still in the nucleus right now because DNA is the nucleus. DNA does not leave the nucleus. We're still in the nucleus. We haven't left the nucleus yet. So all of transcription happens in the nucleus. In eukaryotic organisms, RNA will be processed before it leaves the nucleus. So that gene might contain certain sequences of bases. Maybe there's 50 base pairs in there that are not necessary for building this specific protein. Ever heard the idea that there's a lot of junk in our DNA? There's a lot of unnecessary information. That's kind of what this is related to. There are pieces of our DNA that do not contain or are not relevant for building that specific protein embedded within the genes. So we don't need those pieces. We want to take those out. We want to trim those out. So we trim out those pieces that do not contain the information for actually building that protein. And then we connect together the pieces that do have that information that we need for building that protein. So this whole process of taking out those pieces that we don't need and connecting back together those pieces that we do need for how to build those proteins is called RNA processing, or you'll often hear it also called splicing, RNA splicing. So splicing just means connecting together pieces of a nucleotide. So the pieces that get removed, those pieces that we don't need, that contain information that we don't need to build this protein, we call those introns. So introns are sequences of DNA that then get transcribed into RNA that do not contain information for how to build that protein. They are going to be removed from that molecule. The remaining pieces that we do want to keep in that do contain information for how to build this protein are called exons. So these exons, these are information that we need to build this specific protein. We're going to take out the introns and then connect together the exons. Okay. Again, this is all happening in the nucleus. This is all happening in the nucleus. So this is done by an enzyme. Okay, so there's an enzyme complex, multiple enzymes working together in the nucleus that will take out the introns and splice together the exons. So now we have only exons. Exons are the ones that contain information that we need. So the other thing that's done to RNA before it leaves the nucleus, besides removing introns and splicing together exons, is we're going to add some pieces to it. We're going to add on what's called a 5' G cap. So these are just guanines right here. So we add on a sequence of guanines on the 5' end. On the 3' end, we add on a sequence of adenines. A polyetail. So these have a couple functions. This 5' G cap helps the ribosome attach to this mRNA. Remember, ribosomes is the ones that actually build protein, and they use this mRNA as instructions for how to build that protein. So this 5' cap helps that mRNA attach to the ribosome. This poly-A tail over here helps slow down the degradation of this RNA in the cytoplasm. So we have enzymes in our cytoplasm that are constantly zooming around that are breaking down RNA because we don't want this RNA to stick around forever. We need to reuse these nucleotides, and in a day or two we might not need this protein anymore. So this poly-A tail basically extends the lifetime of this RNA molecule, so that way it can hang around at least long enough to be used to build that protein. Whew. So this is all done in the nucleus. Before we even get to building the protein, we remove the introns and we connect together the exons. And now, after we've done that, and we add that 5' G cap and that 3' poly-A tail, Now we can call this molecule mRNA, messenger RNA. Now that it's been kind of put the final touches on it with these edits to it, we can call it messenger RNA. And this is what will leave the nucleus. This will go through that nuclear envelope, through those pores in the nuclear envelope, head out to the rough ER, find a ribosome there, or it'll head out to the cytoplasm and find a ribosome there. So this is what's used now to actually build a protein. This is our official copy of that DNA sequence that we needed to use.